Hey everybody, hope you're all doing all right. I'm going to give a update here on the exam, and thank you, Richard, for emailing me because I had the exam up, but apparently I forgot to hit the publish button. So it is available now, that midterm exam, and I originally had it due November 1st. I'm just going to push that back since it wasn't available to you guys. You do have to download that Respondus lockdown browser for it. So just want to make everybody aware of that. Also, there was a question about the crop project as well, which is also due either the 1st or the 2nd. I think it's the 2nd. Either way, for that crop project, it was asked if you had to do a recording. You don't have to do a recording, it's preferred, but as long as you hit the checklist up, make sure you have everything covered and well explained. You can either do that through the slides themselves or the notes portion of the slide. If you're doing a slideshow, as long as you cover those bases, that's all right. I'm still going to add that little narration part to presentation I was sharing with you on eggplant, and kind of show you how that's done. And kind of the second phase of the crop project, we'll do some sort of narration, but you don't have to do that for the first one. Just wanted to make that clear to everybody. We will get back to chapter 9, and we left off talking about biotechnology, and just wanted to kind of continue there on the biotechnology front with some updated figures first. So this is from last year, as the book said, and sometimes this edition of the book is a little outdated. So this is back in 2011. They were using 2008 figures. They were saying 86% of cotton, 80% of corn, so on and so forth. So we can see here in this image that's gone up to 96% of cotton, 95% of sugar beets, 94% soybeans, 93% of corn. Again, what we're seeing here is that our genetically engineered crops are those that are mostly just the highly subsidized kind of commodity crops, the field crops. What we think of as more the agronomic crops versus row crops, with some exceptions. I mean, like in here you see sugar beets is on this, or well, sugar beets as well as summer squash. Sugar beets is a field crop. Summer squash, though, is on here. And these letters are just saying HT for herbicide tolerant, VR, virus resistant, IR, insect resistant. That's the one where they put the bacteria, Bacillus thuringiensis, in the actual cells of the plant. DT is drought tolerant. So that's what our crops have been genetically engineered for so far. And these are really the main crops that are out there. They have introduced some genetic engineering technology, some biotechnology into apples as well. But I don't think that's hit the market yet. I could be wrong about that. And of course, we're thinking plant crops here. They have done some genetic engineering of animals as well. Popular case with salmon, where they've figured out a way to kind of rework the growth hormone so that you can produce a full-grown salmon ready for fillet in months versus years. So nonetheless, biotechnology is one of these tools that we use for crop production. And I wanted to, again, emphasize that genetic engineering, I feel like, is the better term to use, because really we have been genetically modifying organisms just through breeding, through hybridization, really. I mean, with hybridization, we're taking two very different crops, crop varieties, or even species in some cases, and crossing them, and we get hybrid vigor as a result from that. And that's a result of new genetic combinations, new trait combinations that perhaps weren't there. I mean, they were kind of hidden in the gene pool, but they weren't there before. So um, genetic engineering, though, is a lab science. You know, it has to be done in the lab, and you're actually inserting genes most of the time from another organism. And sometimes it's from the same plant species or the same plant family. Sometimes it's from a totally different organism altogether. So genetic engineering, I feel like, is totally different than genetic modification, even though in kind of the modern lexicon, the terms are synonymous. All right, so let's move away from biotechnology here, and let's talk about propagation, which propagation is just the process of increasing plants, and either sexually via seed and just kind of taking advantage of the natural processes, or asexually, which we think of as vegetative reproduction. And with asexual propagation, we are basically making a clone of the plant. And in some cases, we are utilizing a plant's natural, kind of taking advantage of its natural reproduction with vegetative propagation as well, especially with particular methods of propagation. And we'll look at these in more detail, but here's just a kind of a list of common methods of plant propagation. Really what we're doing with propagating plants is just trying to obtain and maintain the desirable characteristics of the plant. And so again, with asexual propagation, we might have started with one plant, and then we've cloned them, we've asexually propagated them to where we have, in some cases, maybe even millions of individual plants out there that genetically are a clone. They're the same as the original. And here are just some examples of those, like the Bing cherry, for instance. Fruit trees most often have to be asexually propagated to maintain characteristics because they are highly outcrossing. But some things on here, like the Pawnee variety of wheat or Ranger alfalfa, not necessarily things that you would think of that needed to be cloned, but again, they can be highly outcrossing. In this case, wheat being wind pollinated, alfalfa being insect pollinated. So to keep those characteristics and keep producing the correct seed, you have to have clones. So we use propagation to increase populations, whether those genetics are the same in asexual or different in sexual propagation. If a plant group is actually reproducing true to type by seeds, then we consider a cultivar a line. And a line is homozygous, and if self-pollinated, the seed propagation is going to yield children, offspring that are similar, not necessarily exactly alike the original plant, but have the same traits. And so in this case, we're using sexual propagation, but we're able to keep them somewhat true to type. This is also known as open pollinated seeds. And that's because they are homozygous. In some cases, they are heterozygous too, but generally they have those dominant traits. They're stable. They've been taken to a generation to where there's not a ton of difference in that gene pool. It's kind of more of a narrow gene pool with open pollination. However, they can outcross in that case. So a lot of our, most economic plants, like our grains and our vegetables, are created from seed bred lines. However, many of our forestry species they might be seed propagated. They're not considered lines though, because of their higher level of variability. You can also look at the inbred lines that are used to produce the hybrid cultivated varieties, and then hybrids themselves, which very often in corn, two different parents combine to get this more vigorous plant. In a lot of cases, these are heterozygous, so they have many dissimilar genes controlling their characteristics. And as we talked about on Monday, sometimes it takes a lot of genes just to code for one particular characteristic, like color, for instance. They all have to be right in order to get that color. So in that case, we have heterozygosity, but we can still get plants and progeny that are similar to the parents. If we want to maintain it exactly, though, we do vegetative or asexual propagation, and that yields a clone, a genetic clone. It's kind of funny to think about how, I guess it was back in the early 2000s when there was such an uproar over cloning, when really in the plant world, we've been doing it for, really thousands of years in some cases. So if we are trying to maintain that cultivar through seed propagation, through asexual propagation or sexual propagation, sorry, I keep getting those mixed up, we have to very carefully control the parentage and the source of the seeds. So one of the ways we do that is by separating or isolating the plants either in space or time, and that will prevent crossbreeding, pollen contamination, and the loss of that genetic purity. And so I say isolated in space or time for our seed saving, because there are a couple of different ways you could do it. In space, say for like peppers, for instance, they say that you want to separate your sweet peppers from your hot peppers by a minimum of 25 feet, because peppers are usually self-pollinating. However, they can cross-pollinate. So keeping them separate and by that amount of space, then they're able to stay pure. You can also do it in time. So for instance, say you're saving a sweet corn and maybe a field corn variety. They're going to actually mature their flowers at different times. So most sweet corns, 60 to 70 days. Most of our field corns, grown more for like meal or roasting ears, are around 100 plus days. So they're not going to be flowering at the same time. And corn pollen doesn't last that long. So that can prevent that loss of genetic purity there as well. So isolation is kind of how we get around that with asexual propagation. However, with certain plants that are propagated by seed, we might want that variability. In some cases, we need to have that variability because some of these plants have such high inbreeding depression that if they're constantly inbred, they will die. So where we want plants to adapt is really where we want to get some of that variability in there. And I think if you're seed saving at home, you want a little bit of variability, because in doing so, you're actually kind of doing some plant breeding just through selecting a particular parent that has done better in your garden than the others. In doing so, you're creating something that's more adapted to your garden or your farm. So I think everybody really should participate in seed saving and plant breeding to some degree. It's not that difficult, and we can get some excellent results from it. Most countries and states have some sort of seed certification programs that are protecting and maintaining genetic quality of different cultivated varieties. So often, whatever government agency it is, usually at the state level, there are some federal regulations as well, basically set forth these standards for the quality and the characteristics they want. And this is known as certified seed. And there are different certifications of this seed in terms of genetic purity or free from weed seed. often both. For like seed potatoes, which is different because it's asexual, it's cloning it because you're doing it vegetatively rather than reproductively there. With certified seed potatoes, it's a little bit different. It's virus free in that case. Either way, it's different for each plant, but there are some standards out there often maintained by government agencies, different categories of certification as well. And most farmers in North America do use these certified seed from recognized producers. So let's talk about seeds for a second and a little bit of review here. Of course, the seed has the three essential parts: the embryo, which is that live part of the seed developing into the new plant. We've got the food storage material that feeds that embryo, and then some sort of covering of the seed, a coating of the seed. And for a seed to germinate, that embryo does have to be alive, but then we also have to get the correct environmental conditions in order to get it to actually germinate. So there are a bunch of different tests that we can do to actually, see whether it is viable or not to a certain degree. And one of these really simple tests is just to float seeds in water. And usually the ones that float to the top don't have a living embryo on them, and those can be skimmed off because they're no good. So that's a good simple test you can do to see whether they're good. That doesn't work for every seed because some seeds are buoyant and do float even if they do have an embryo living inside of it. Germination test we often use basically just to get a certain number of seeds, and we calculate the percentage of the seedlings that develop from the number of seeds planted. So again, another simple one, but it requires a lot of seeds to do it. They want you to do it, and the way that they do it actually for like seed companies is they'll get lots of 100 seeds for each lot, and then test each lot, and then if the percentage differs amongst the lots by more than, I think it's like 5%, then they do it all over. So if you're a seed saver, you want to sell seeds, which I would highly recommend as kind of a supplemental source of income on the farm. They're in high demand, especially organic seeds, because they have to have them for organic crop production and organic certification. So I would recommend it, but nonetheless, they really want you to do a germination test before you send it in, because they're going to do it. And if it doesn't have good germination, then they're not going to pay you well for your seeds at all. There are other tests as well as the typical germination tests, such as a tetrazoleum test, which uses a chemical that reacts to living tissue. An excised embryo test, where they actually take that embryo out, and that doesn't just look at whether it will germinate or not, but also looks at how viable and vigorous the seedlings are. There's also X-ray tests too that they use actually to certify a lot of seeds. Seeds are incredible, really, because they are just dormant. I mean, they're living, but they're able to go dormant. It's the way that an annual continues its life cycle beyond that one growing season, right? So anyway, this dormancy is an important survival mechanism for many flowering plants, and it is a result of evolutionary development. And we have to overcome that dormancy in order to get our seeds to germinate. So how do we do that? Well, one, we look at the actual seed coat dormancy, which the seed coat is just that outer part that makes it impermeable to water and gases so that it doesn't mess up the embryo on the inside. So we use methods of softening or altering those seed coats, in some cases completely removing them. And we refer to all these methods as scarification. So scarification is the physical or chemical treatment of a seed to remove that seed coat. So that could be through like rubbing it with a file or sandpaper. It could be using heat treatment, or even using an acid like sulfuric or a lot of times hydrochloric acid, and that simulates the passage of the seed through an animal's digestive system. Um, Again, with these, overcoming seed dormancy, we have to do this really in some cases to get better germination. So this happens in nature, and of course in nature, a lot of plants are producing tons and tons of seeds, so that just a few can end up growing and germinating and being another parent plant, passing on that DNA. But if we scarify them or use other techniques, then we'll increase that rate of germination. So another way is like with tomato seeds, we ferment the pulp, and that fermentation, which makes it easier for the pulp to come off of the seed and release it, but it also removes some of that seed coat dormancy too. So a lot of different ways to look at scarification. Happens in nature, and we kind of simulate that in order to get seeds to germinate better. Embryo dormancy is just where the embryo does not get ready, or it's blocked from germinating from some sort of growth inhibitor, often growth hormones. And we overcome this through stratification. Stratification is basically just keeping seeds in a cool, moist area for whatever period of time it is. Don't want it too moist because it has to have the right amount of oxygen as well. But what they believe happens is this kind of after ripening of the seeds and the embryo within the seeds. So it could be increasing the growth promoting hormones and decreasing the growth inhibiting hormones through this process. Or it could just be, allowing that embryo to further mature. So either way, stratification is often looked at a number of days, often putting seeds like in a freezer or kind of simulating winter. And almost all of our native perennials have to be stratified in order to get them to germinate in decent numbers. There are some species out there that have both a seed coat and embryo dormancy, so they have to go through some sort of stratification and scarification. So this double dormancy is, in some cases, might even take several seasons or several years to overcome. So let's look at just the sequence of events for seed germination. Starts with imbibition, taking up water. Then it moves to activation, where basically that water is used to activate various enzymes and change up the hormones inside of the seed. And then we get that embryo, the embryo actually growing and developing, breaking through the seed and seed coat and emerging. And of course, we have the cotyledons, which are our seed leaves that are kind of feeding that until it can photosynthesize on its own. And usually those cotyledons will drop off after it's emerged and the first true leaves appear and can start photosynthesizing. So what are the environmental factors that influence germination? Of course, moisture, not too much, not too little. Temperature is one of the main things, especially with annual plants. Needs to be warm enough. Our warm season crops, we're talking usually mostly over 60 degrees before they can germinate. So we might put them on like a heating pad, for instance, to get them started early. If we're transplanting them. They need a certain amount of oxygen as well, so they can respire. And in some cases, they need light, like lettuce or celery, a lot of grasses, they need to be on the soil surface and not covered. Um, in some cases, they need an absence of light. We also need freedom from pathogens, and seedlings can easily be killed from diseases like damping off, which are caused by a variety of fungi and oomycetes. Also, freedom from toxic amounts of salts, because salts can inhibit the whole moisture concept. So sometimes over irrigated soils can reduce seed germination as well. All right, so that's the sexual propagation. We actually develop seeds, we use them. Now let's talk about asexual propagation. Of course, in the sexual world, we were talking about how we go through meiosis. But of course, to get to that point, the plant has to grow vegetatively, and it's doing that through mitosis. So a non-reductive cell division where the chromosome numbers stay the same. And basically the cells are just creating copies of themselves, and we use that to vegetatively propagate. And what we need to do is basically create shoots and roots. And different plants, one might come first. So usually we think of creating the roots first, getting it to root before it can develop new shoots. Both of these occur as a result of callusing. And a callus is just this collection of these, what are called parenchyma cells, that are healing the wound. Say when you make a cutting, for instance, or you make a graft, these parenchyma cells create this callus. From that callus comes the roots in many cases, not all of them. Adventitious shoots are those that appear any place other than where they're expected to come from, the shoot terminus, or in the axils of the leaves. Adventitious roots are the same deal, except again, they're roots coming from any place other than where you would expect them to come from, underground. So adventitious just means that it's able to appear where it's really not expected.
I have a question.
Oh yeah, go ahead.
Is that what potatoes do when you leave them in the dark, like pantry for too long, and all those little sprouts come out? Is that what that's doing?
Yeah, basically we're getting both adventitious shoots and adventitious roots occurring when potatoes are, and usually that's kind of happening because potatoes are basically clones. We're pretty much taking that tuber, which is technically a stem, part of the stem, and when we remove it, we're and we cut them into pieces, we're technically making little cuttings. So they do reproduce by that naturally, as well as we kind of will get them to sprout, sometimes intentionally to get them to produce quicker. For eating purposes, though, we want to try to prevent them from sprouting. And once they've kind of been in a place long enough, usually in kind of more humid conditions, then they will sprout. Or if they have enough light, they'll sprout and make those adventitious roots and shoots. So here's a picture of a callus here in a grapevine. And callus is a result of wounding. It's just this mass of undifferentiated parenchyma cells. And that's usually where the roots come from, especially in the woody plants. Some herbaceous plants, you don't necessarily have to have that. So we use cloning or vegetative propagation for plants that are highly heterozygous, or those that do not breed true from seed. Or if we have a mutation, then we try to preserve it through cloning. One way that we might combine two different kind of genotypes together is through grafting. And that's where we use a scion, which is the top part of the plant, as well as a rootstock. And that's the bottom part of the plant, making the root system. and they get characteristics from each. So some cases, like for apples, for instance, we're always grafting apples, most fruit trees actually, because the root stock um, will impart some disease resistance or hardiness, and then the scion is the apple that we want to eat. So more about grafting here in just a second. Almost all of our fruit and nut cultivars and many ornamentals are clones. And we have to recognize in that whole cloning thing that we're getting that genetic uniformity. So genotype allows them to potentially all be the same. However, we can't forget the phenotype, how the environment interacts with that genotype to actually create the plant's appearance. So cultivated clones actually are going to release either as seedling plants that are propagated vegetatively or from mutations. And we call these often bud sports, where we have a single cell that has a different genetic makeup during cell division. And so often that will happen just like on one branch. So that's how we've actually gotten a lot of new varieties of fruit trees. It just mutated on one branch, and then we vegetatively reproduced that. And as I mentioned, again, the environmental factors can modify the appearance and behavior of a plant. So we might have a mutation, we might try to preserve it, but it might not always stay exactly the same depending on what the environment is, or if it further mutates. Also, we have some mutations that genetically change just a very small portion of part of a plant rather than the whole shoot. And these are called chimeras, like the mythical beast that had like the head of a lion and the tail of a dragon. So we see a lot of different patterns in chimeras, and often like variegation. Like in this case, this variegated pink Eureka lemon is a type of chimera. So we get some pretty cool stuff with chimeras, but it's not always easy to reproduce chimeras. And they actually categorize these chimeric plants based on the location of the mutated cells. So in a mericlinal cell, it's not actually happening kind of on the what we think of as the growth plate. So it's only just a region or portion of the plant, not as easy to kind of maintain that genetic mutation. Same with sectorial. It's only happening on a section, and it might go from the growth plate into the meristem, but it's only on a particular section. What we really want is a periclinal chimera. These are the most important because they are relatively stable and can easily be vegetatively propagated because it is positioned near that apical dome where the cells are dividing. The other chimeras don't often, they're not as stable. So not to say that they can't be reproduced, but it's just not as easy to do so. Of course, we also have problems in clones with passing on disease. So if we have viruses, we're taking a disease or virus infected part of the plant, then usually it will be carried over unless we remove it. And often that's through some sort of heat or chemical treatment. Another way to do it is through this shoot tip culture, which we'll talk about here in a minute when we get into tissue culture. So let's go into the simple parts of plant propagation. Cutting's probably the most simple. I'm sure everybody's take a point or another. And I'm going to put up a lab asking you guys to take a cutting if you haven't already. And I'm going to send you guys some material to take cuttings from. So cuttings can be classified according to the part of the plant from which you get them. And we have stem cuttings, leaf bud cuttings, and root cuttings. Again, all from different parts of the plant and taken at different times of the year, different growth. There's a lot of information out there about how to propagate plants, what type of cutting is best for what type of plant. However, it's not all out there. So there's a fair amount of just trial and error of things that you all need to experiment with in order to propagate it. I guess the wealth of knowledge is getting better on the web, but it's not all out there, especially even when we talk about some of our native plants that really haven't been propagated all that much. Grafting and budding are kind of synonymous. Again, with grafting, we're combining two different parts of a plant, the scion and the rootstock, in order to get the plants united to where they're growing as one. And in some cases, we might have multiple varieties growing together on one rootstock. You might have seen that. I'll put up a link too of one, this project, they call it the tree of 40 fruits or something like that, where they put all these different fruit producing or nut producing plants that are in the same family, the rose family, all on one plant. So you could get like apricots, peaches, plums, cherries, almonds, kind of all on one plant, or apples and pears could be on the same plant. Various types of citrus. They do have to be closely related for grafting to work, though. So there's multiple types of grafting as well. Root grafting, or top working, which is used to change a whole cultivar of a tree. We also have bridge grafting or in-arching, which is used mostly to repair damaged tissues of a tree. And we also use sometimes to study the transmission of viral diseases, and that's called indexing. So here's just a look at some of the simple types of grafting. Whip grafting is the most common, where basically we're making multiple cuts in your rootstock and your stem, kind of matching angles, but then also adding, in this case, whip and tongue. So you actually cut it in the center so that you get this groove, kind of like you would with the tongue and groove carpentry, so that they fit together and hold together well. So picture like this, they're interlocking there. And the big thing with grafting is that we have good matchup of the cambium, the vascular cambium itself. That's what allows them to grow. So generally we want our scion and rootstock to be about the same diameter, and usually fairly small too. And often we will either tie it or wax it, or sometimes do both. Usually parafilm is, or buddy tape are the two that are most often used for whip and tongue grafting. Another form of grafting, which we use for top working, is cleft grafting, and you can take like a cleaver or this particular cleft grafting tool. Use a mallet to basically split open the top of tree. You cut the top off and you split open the trunk, and then you insert the scions in that edge. So kind of like this. And eventually those will grow. Typically we want to wax the top of that as well. And the waxing is used to prevent moisture loss, keep them moist enough to where they do unite and grow as one, but also to keep insects and disease out as well. So more types of grafting. Budding is also very common, and this is where we're just inserting a bud instead of a whole branch or a whole scion. And With the other types of grafting, the important thing is that your scion wood needs to be dormant. So usually you're collecting those in the winter time, and grafting them before they wake up. You'll put those often in a refrigerator, keep them moist, until you have the right time to get out there and graft. Usually when it's starting to warm up a little bit. Budding, however, can be used pretty much anytime. You can do this in the spring or in the summer. So that's one reason why it's, I guess, more popular in some cases. And for some plants, you have to do it that way. So tea budding is one of the most common, and it's called tea budding because the cut is in the shape of a T. One of the most important things for grafting, the compatibility and making sure that your scion wood is dormant, is just the union, that contact of the vascular cambium and the callus that forms around them, this process of healing, which usually takes a few weeks, and it has to be completed before the buds on the scion start to grow and develop. So that's the reason why we're doing this with dormant scion, and often the reason why we're doing it in the cooler parts of the year. So timing is of the essence with grafting. And here's a look at healing for cleft grafting. We've made that cleft, and then we've put in our scions so that that vascular cambium matches up and makes a complete ring. And then basically what's going to happen is that callus is going to start to form and kind of bridge with these parenchyma cells to cover it and allow these to intermingle and eventually interlock to where they can produce new xylem and new phloem and become a one plant. So again, they have to be compatible, both the rootstock and the scion, and that's looked at as some degree of botanical relationship. Generally, the closer, the better. You cannot graft things out of the family level, but you can graft things within the family, or sometimes between different genera. So in this case, we can get an intergeneric combination, as pictured here, kind of they say a quince and pear, but I'm kind of thinking that this is actually looks like more like a crab apple to me. Either way, we can get some combinations here where they work, even being different genera. It's most able to be done when they're the same species, or even if they're clones of each other. If they have the same genetics, then that's a really good way to practice and build confidence on grafting. One of the things that they often do for grafting, especially for tree fruit growers, is graft onto a dwarf or semi-dwarf rootstock so that harvesting is easier. Yeah, so that's very common, especially for backyard fruit growers. But that rootstock can have all kinds of different influences on the scion itself. In the case of citrus, it can even kind of change the quality of fruit. We also start to do, I mean, we've been talking mostly about woody plants, but we have been doing some herbaceous grafting as well. Especially because methyl bromide, which was once used as a soil fumigant, was banned because of its negative health effects. So a lot of times, especially now in the world of tomatoes, we're using kind of virus or disease resistant rootstock, and then maybe like a flavorful variety or a more vigorous variety of scion. They do that with squash as well now. A lot of plants doing more herbaceous grafting now, especially as we're creating more and more tools to help with that grafting. Another type of propagation, vegetative propagation, is layering. This is mostly done with, I guess, plants that have flexible stems. So tip layering is done actually just burying the tip. That's really only done for like cane fruits like raspberries and blackberries. They have the ability to sprout from that tip. Very easy way to propagate them. They do it in nature as well. Try it out. Great way to get more blackberries or raspberries. Mound layering is often used, this is also called stool layering too, for making rootstocks for fruit trees. So what they're doing here is they're basically taking a parent plant, they're going to top it, they're going to cover it with soil or some other kind of media, and it's going to sprout and produce roots. And then we take those become our rootstocks. Quick way to produce rootstocks. The most simple form of layering is just taking a flexible stem and just burying it and letting it produce roots. In this case, they have it staked down. You can also kind of, nick it, cut into it in order to get it to produce roots. Often through wounding, we induce rooting. And yeah, then once it's produced roots and we start to see some new growth on that shoot, then we just dig it and separate it from the plant. Air layering is another type. This is happening on the plant. We will strip the bark off and wrap it around with some sort of media, often like a sphagnum moss or some moist moss. Wrap it in plastic and then watch the roots form. Typically we girdle that stem through cutting it, and then we'll maybe put some root promoting hormone, some auxin powder rubbed onto it. Auxin, again, we talked about it a little bit, is one of those hormones that is used to promote rooting. So in this case, we're wrapping sphagnum moss around that cut and then using plastic or aluminum foil to wrap it around, and then getting those roots produced, and then just removing it from that plant and planting it on its own. Some plants are able to just grow from the stolons, these kind of runner type stems that come from the crown. And these new plants that arrive at the nodes are kind of a natural way to just take advantage of layering. So for strawberries, that's a very common way to do those. In other cases, we're using adventitious roots that just develop from a rhizome or horizontal root system, which spread to form a thick, dense thicket. And in those cases, we're just cutting that away from the parent plant and replanting it. So as long as it has some roots and some shoots, usually it will grow. We call this division if we're manually cutting it, or we call it separation if we're using the kind of natural form of the plant. So think of using things like bulbs and tubers, like potatoes or garlic. We take these little baby parts and just separate them from the mother plant. We're not actually cutting them away. So that's separation. Division, we're actually manually cutting it. So with bulbs, we're often, using that kind of that main form, these side bulbils that form as a part of it. For things like bamboo and irises, we use that rhizome, and we divide it from the other rhizomes and then plant it and it grows. Again, as long as it has a shoot and a root, usually it will grow on its own. So we're taking advantage of these specialized stems and roots as a way to propagate a lot of plants, and some of these are economically important, mostly more in kind of tropical regions in terms of fruit producing plants like bananas and ginger. But for the ornamentals like ferns and many of our grasses, that's how we do it. You can easily grow ferns and a lot of grasses from just dividing them from the other part. I know we're just a little over time, and I just have one more thing to share with you guys before we sign off. If I can be a pirate for a second. and that is tissue culture. So micropropagation or tissue culture is basically just growing a plant from any piece or part of the plant that can be used to regenerate a new shoot and root system. And we're growing it under aseptic conditions. So we're growing plants in test tubes basically with tissue culture. And as I mentioned before, that's also a way that we can sterilize parts of plants and get them virus free. So this is done in labs usually, but you can, I mean, it can be done even in a kitchen. So generally we're just taking even just one cell of a plant and then putting it on this nutrient medium, often kind of like gelatin. It's like an agar media. We have to sterilize that stuff because that nutrient media is also favorable for the growth of things like bacteria, fungi, and yeast. So we sterilize it, and then we basically give it everything that it needs, including mineral salts, you know, basically the minerals that the plants would get from the soil otherwise. We also give them vitamins, the sugars that they would need through photosynthesis, we're applying to that media. And then we also give them the proper growth hormones or regulators in order to get them to form shoots and roots. And that is looked at usually as a ratio of the auxin to cytokinin. Either way, we add these to that nutrient media that they grow on, and then we subculture them, continually add those media or change it out. And eventually when they have roots and shoots, then we'll grow them in a protected area for a particular amount of time, and then eventually move them on to soil and become a new plant. All right, so last thing here on micropropagation, table showing the components of a particular type of commonly used medium for tissue culture. So you can see this is basically everything that you put in it. There's often different formulas for different plants, and this is also another level where the science is out there in some cases, but it also needs to be further experimented with to actually, for a lot of different plants, because it's only the most common plants that have been propagated using this method. All right, and that covers the end of our section there on genetics and propagation. I've got that quiz up if you want to take that at any point. If you guys have any issues with the midterm or the crop project, just give me a shout. Let me know and we'll work it out.
Okay.
Otherwise, hope you guys have a great day.
I hope we do too. Bye, everybody.
Talk to you soon.